One main thing about all amino acids is that they are chiral. I mentioned that this is a central carbon with 4 different groups attached to it. And remember, that is the definition of having a chiral center. So amino acids are chiral, except for one of them, and that is glycine. Why is glycine not chiral? If you remember, glycine's R group is a hydrogen. So if you have 2 hydrogens attached to your central carbon, you do not have 4 unique groups because there are 2 groups that are the same. So glycine is not a chiral amino acid, and that is the reason why. Otherwise, all amino acids have chirality. That means that they could exist as what we refer to in organic chemistry, the R or the S configuration, right? And that has to do with the way that each of the 4 groups are positioned around the central carbon. Another way to talk about this is the system that we use in biochemistry, which is the D and L formation. Generally, D and L are going to relate back to R and S, where our R will be related to D and our S will be related to L. These abbreviations come from the ability of the particular biological molecule to rotate light. This is going to be in a right-handed or left-handed rotation of light. Now, for amino acids, we say that amino acids have a L formation in all biological systems. So naturally, natural amino acids within our body and almost all living organisms that are utilizing amino acids are in the L formation. Now, that is outside of a few organisms who naturally use D amino acids, like certain bacteria. But know that the form of amino acids, all of our proteins, only contain the L formation of amino acids. And this stereochemistry is important because the protein building blocks will not accept D forms of amino acids. And just for binding pockets and areas of our body will not be compatible with the simple restructuring of the amino acid in the D form, in the other stereo form, the other enantiomer. Now, aside from each amino acid having its own special R group, which we have gone over, the amino acids can be placed into 5 different groups that I will be defining here, depending on what type of R group they have. We call the amino acids by their groups of either being in the non-polar group. This means that they have an R group that is simply non-polar. It can also be put into a polar group. This is polar and uncharged, so it does not have a charge on it, but it is polar. Most of the polar R groups can hydrogen bond. Now, there are also 3 amino acids that have a positive charge on them, so they are in the positively charged group. There also are 2 amino acids in the negatively charged group. Sometimes, depending on texts you are reading, these will be lumped together into one group that is just called the charged amino acids group. They both have charges. Half of them are positive, half of them are negative. Here we'll be defining them as their own separate group. And lastly, there are the amino acids that are aromatic. These have some type of aromaticity, and they can exist, again, in different forms based on their aromaticity. First, we're just going to be going over those amino acids that we looked at and showing you which group they fit into. First are the non-polar amino acids. These include glycine, alanine, proline, valine, leucine, isoleucine, and methionine. Now, notice that generally these amino acids, their R group is all composed of just carbons and hydrogens, except for methionine, which has a sulfur. There are no charges on here, and there are no elements of oxygen or nitrogen or even sulfur that have extra lone pairs that are going to be able to make them potentially charged. The next group I'm going to cover quickly is the aromatic groups. These are mixed between being polar and non-polar, but the general grouping part about them is that they have an aromatic ring. So notice all three of these have some type of aromatic ring, a benzene ring within their structure. This includes phenylalanine, tyrosine, and tryptophan. All three of these have a special ability in that they can absorb UV light between 270 and 280 nanometers. And again, that is because of this aromatic ring that is present. The next group is the polar group, but uncharged. So remember, these do not have a full charge on them, but they are polar. They have some partial ability to be charged. Most of them are going to be able to form hydrogen bonds as well. So this includes serine. Notice it has an OH, an alcohol group on it that makes it polar. Same with threonine. Asparagine and glutamine are going to have these amide groups that make them very polar. But again, they don't have a charge on them. And then lastly, cysteine has this thiol group, again, that is separated. It's not cloistered within carbons and hydrogens. It's at the end of the group. This makes it very polar. And in particular, cysteine is going to make disulfide bonds. So review that functional group, the disulfide, when we have a disulfide bond, and its relation to cysteine. Cysteine is very important because, and it's very unique because it solely is going to be able to make this special kind of bond. And we're going to really talk about it a lot more when we get into 3D structures of proteins. But remember that cysteine is polar and that it can make these disulfide bonds. The next group is going to be the positively charged amino acids. So again, this includes lysine with that NH3 plus group, arginine, which has that NH2 plus group, and histidine, which, if you remember, in this case, the nitrogen on the ring that is double bonded has this NH group and is generally positively charged. Next is the negatively charged R groups. So aspartate, again, we can say that as aspartic acid, and glutamate or glutamic acid. both have these carboxylic acid groups at the end, which make them negatively charged. Okay, so those are the main 5 groups that we've gone over that we will be grouping our amino acids into based on their special R group.